Hi everyone, today we're going to be discussing Chapter 4 from your textbook, and Chapter 4 is going to be covering the functional anatomy of prokaryotic and eukaryotic cells. Big takeaways from today's lecture are going to be comparing and contrasting these two cell types, and there's also going to be a pretty big focus on features of prokaryotic cells because this is microbiology, so we'll be discussing the different types. All right, so for just to get some definitions out of the way, we have prokaryotic. The pro here is talking about pre-nucleus. And then eukaryotic cells, the root there means true. Whenever you see the term kary, like a karyotype or a karyote, it's talking about like a chromosome or like a nucleus structure. And just to orient you guys here, so this cell is actually a really enlarged version of a prokaryotic cell. It doesn't have a real nucleus, but it has genetic material here, right? So its chromosome is circular and it's just kind of floating around in the cytoplasm. It has some ribosomes, but no other organelles. So we're going to talk about the features it does have from kind of an outside-in approach. So we're going to start with all these fun extracellular structures and then work our way in. And this guy over here is a eukaryotic cell, and we have plant cells over here on the left, and our animal cell little diagram over here on the right. So there are types of microorganisms that are composed of animal cells. We'll talk about them as the class progresses. But, just keep in mind, we also are composed of animal cells. And there are also microorganisms that are plant cell-based, and we'll discuss them a little bit later. But our primary focus of this course will be on bacteria and other eukaryotic protists and protozoa that cause disease. So, all right, let's get into it. So this slide is good. I like it. It's a good compare and contrast, prokaryotic cells versus eukaryotic cells. So prokaryotic cells have one chromosome, and it is circular in nature. But as we saw on the last slide, just to go back, just because it's a circle doesn't mean it will always look like this, right? So if it's a really big circle and it starts to get coiled up, technically I connected the ends. This is still a circle, so you can imagine that's what's happening here in the cytoplasm of our bacterial cell. So they do have genetic material, they do have DNA, and they do have a chromosome, but there's no nucleus, right? We'll talk about it in a second. The area that their genetic material is found in is actually called the nucleoid. So no nuclear membrane, but they do have their material kind of in the same spot called a nucleoid. They don't contain any organelles, but we will talk about what they do have in the cytoplasm. And then this term might be new for you guys, peptidoglycan. This is a type of protein sugar molecule that is found specifically on the bacterial cell walls. It's important when we talk about different types of bacteria, how much peptidoglycan they have and where it's found. So we'll talk about gram-positive and gram-negative bacteria. And they reproduce via a process called binary fission. So it's similar to mitosis in that you start with one cell and you end up with two, but there are a lot of differences that we'll highlight as we go through the lecture. All right, so eukaryotic cells have multiple pairs of chromosomes. So, for example, humans have 46 pairs of chromosomes, right? You get half from your mom and half from your dad. And those chromosomes are found in the nucleus, so there is a nuclear membrane. They have lots of different organelles that we'll cover. And again, this should be a review from general biology, right, like mitochondria, smooth and rough endoplasmic reticulum, etc. If it is a plant cells are eukaryotic. They have cell walls, but their cell walls are composed of more complex sugars or polysaccharides as opposed to peptidoglycan, which is primarily found or only found in bacteria. Okay? All right. Eukaryotic cells also reproduce via mitosis, which is a specialized type of cell division I'll touch on very briefly, but not going to dive into it too deep. You should have had that in general biology. All right. So one of the big differences between prokaryotic and eukaryotic cells is the size of the cell. So the average size for prokaryotic cells is going to be much, much smaller generally than eukaryotic cells. This term right here is micrometer. It's the Greek letter mu, so that's what you're seeing here. And they don't use an M because mm is for millimeter, right? So micrometer is 1,000 times smaller than a millimeter. So if you think about the thickness of a penny, which is about 1 millimeter, if you could divide that into 1,000 smaller slivers, one of those small thousandths of a sliver would be a micrometer in thickness. So that's about how large bacterial cells are. So again, you wouldn't be able to see them, it's so thin. Okay, this slide, super important to get down. Look at it, review it, study it. These terms you will see repeatedly over the course of the year. These are the terms we use to describe bacterial cell shapes. A bacillus is the term we use to describe a single rod-shaped bacterial cell. If they're multiple, we use the term bacilli. So you'll see an I here instead of a US. So bacillus or bacilli. The next term we come to is coccus, which is describing spherically shaped bacteria. So here we have right, so it's a sphere. Coccus is the term used for a singular, and if there's multiple, we use the term cocci. And then in addition to rods and spheres, bacteria can also come in spiral shapes. So we have a couple different terms we can use to describe those spiral shaped bacteria, spirillum, vibrio, or spirochete. And then you'll also sometimes see coccobacilli bacteria. So those are ones that are going to be more football-y shaped. And so it's not, it's kind of a tweener. So we say it's a coccobacillus. Right. Okay. Some other terms that are used to describe the arrangement and shape of bacterial cells. So if you find under a microscope that the cells tend to be found in pairs, you use the prefix diplo. So I can have pairs of spherical shaped bacteria, so diplococci or diplococcus, or I could have pairs of rod shaped bacteria, so diplobacilli or diplobacillus. They can be found in clusters or groups, kind of like a little, thing of grapes, right? So if this is like the little grape leaf and stem, I don't know. This is a Staphylo arrangement, so Staphylococci or Staphylococcus. And so now this term Staphylococcus might be familiar to some of you because we'll see in a second as we go through the names of the bacteria, the genus and the species names, often they're named for how they look or how they appear. So Staphylococcus aureus, if you were to look at that under a microscope, you would actually see clusters of spherical shaped bacteria. And that's one of the ways we can identify and differentiate Staphylococcus from other types of bacteria. Streptococcus or streptobacilli, again, if you guys have ever had strep throat and you were to look at the bacteria that was causing that infection, you would see under a microscope a chain of spherical shaped bacteria. Right? Think about strep like a strip. So strep. Okay. All right, so here we have our prokaryotic cell, and just we're going to be going through the structures here. We're going to start with extracellular structures and then work our way in. So again, we're going from outside in, describing structures. And just so you guys know how they generate these little diagrams or cartoons, is actually from real life images. So this type of image is taken with a transmission electron microscope, or TEM. So you're actually looking at a slice of the bacteria, so you can see this cell wall. And you can see where the genetic material or nucleoid is for this particular bacteria. And we can see some inclusion bodies here as well. So we'll talk about those in a little bit. But so just so you know, these diagrams that you see in the textbooks are based off of real images that you see. One thing to note here also in the image, this scale bar represents 0.5 micrometers. So that's about the width of the bacteria. It's about half a micrometer. And then maybe it's 1.5 roughly micrometers long. So very small microscope. That's why it's called a microorganism. Welcome to microbiology, right? Okay. So on the very outer surface of our prokaryotic cells, we can have you have bacterial cells that have the potential to have structures that surround them on the outside. So what you're looking at here, let me get my pen. So we have the bacterial cell wall is here in blue, right? And then all on the outside here, so from like here to here, right, is something that this bacteria has secreted. This secretion, right, and so you can see other areas of secretion here, right? This secretion is made out of glycocalyx, right? Again, the bacteria has secreted it. It surrounds itself with it. In most bacteria that we find within the body that cause disease, this glycocalyx organizes itself into a capsule, right? The other option for bacteria that don't live inside human bodies, that live, in the environment or outside, the glycocalyx organizes itself into a slime layer. The slime layer is a little bit more unorganized, but it serves a role for cell attachment. So you can imagine if it's sticking to a rock or something, it would have that slime layer would be useful to help it adhere to the surface. In the human body, the purpose of a capsule is to prevent phagocytosis. So you have components of your immune system that we'll discuss that are actually attracted to little receptors that are on the cell wall of bacteria. And if they find these receptors, if they come into contact with them with an antibody or something, they're immediately phagocytosed and killed. But this capsule, right, is going to prevent this recognition from occurring. Right, so the capsule kind of prevents that antibody from recognizing that surface epitope, and so it kind of can hide out from the immune system that way. So we will see quite a few bacteria that are pathogenic, so that's bacteria that are able to cause disease, will have capsules. And again, that capsule is helping them to evade phagocytosis, which is basically being engulfed by your immune cells. Was there something else I wanted to say about the capsule? Oh, yes. So one of the ways we can combat this defensive mechanism is by immunizing us against proteins and components that are found within the capsule, right? So we'll see a lot of vaccines that help immunize us against certain bacterial infections will actually have components of this capsule, and we're trying to train your immune system to recognize the capsule as, foreign, and so it will eat it. The Hib vaccine, the HIB vaccine, a bunch of vaccines for Streptococcus pneumonia, they have this feature. Okay. All right. So leaving the kind of secreted material behind, we have these kind of more concrete structures, flagella. You may have seen flagella when you guys discussed sperm previously, right? Sperm is a type of eukaryotic cell that has a flagella. So bacteria also have flagella. It's just going to be in a slightly different arrangement than a eukaryotic cell, right? In eukaryotic cells, you have the plasma membrane, and you don't have a cell wall. So the plasma membrane can kind of surround the flagella, and you have plasma membrane on the outside, and you're going to have microtubules that come from the cytoplasm and make the cell, or make the flagella. But here, because we have this very thick cell wall, what the bacterial cells do is they create a long filament, and then they have to have something that anchors it into the interior of the cell. So they have a hook portion, which is going to be curved, and then they have a basal body which anchors it into the plasma membrane. So right, this hook portion. Okay. And so how the bacterial cell actually moves the flagella is by spinning this basal body, this anchoring portion, and by rotating this, you can imagine then you get like the wiggliness of the flagella. So you have the filament, which is the long hair portion, the hook that helps anchor it into the plasma membrane, and then the actual anchor portion that is considered the basal body. You might see this term in anatomy. Basal is talking about basement or anchoring. Basement membrane, basal. Okay. So that's flagella. All right, so if you're a bacteria and you have flagella, you have a couple options. You can just have one flagella, so you look kind of like a little tiny sperm, right? So that would be monotrichous. You can have a little tuft of flagella, so this guy would say has a little tuft of flagella at one end, so he would be lophotrichous. You can have flagella all around the outside, right? If he had flagella all over the place, this guy would be peritrichous. You also have bacteria that can be amphitrichous, so we'll see some spirochetes, right? So if this is a spiral-shaped bacteria, and he has flagella at this end, and he has flagella at this end, he would be considered amphitrichous. And he would be also polar. So this guy would be a lophotrichous polar bacteria. There's a slide coming up. It goes through, it gives you examples of each. So again, this I would say, highlighter. Right, so this guy is peritrichous. He has flagella all around. This guy is lophotrichous and polar, so it's at one end. And there's lots. Monotrichous and polar, so this guy has one on one end, and amphitrichous and polar here, right? And these are his like little tiny flagella. Cool. All right. So that's flagella. And the function of flagella is the same, whether you're talking about a sperm or whether you're talking about a bacteria. The function of flagella is to allow the cell to move, right? It's just a few bacterial cells against the world. So they have to be able to, run away from white blood cells. They have to be able to run towards nutrient sources. And that movement is called chemotaxis, right? So there's a couple really cool... I'll try to post it. There's a little fun video you see of bacteria actually trying to run away from a white blood cell in a human circulation. So it's kind of cool. I'll post that. Bacteria can have two different options in terms of motion. If they have flagella, they can run in a straight line. So a run is like they're kind of all spooling all behind them, like or if they need to change direction, they can kind of spread their flagella out in different directions and tumble. So you can run, and if you need to change direction, you can tumble, run. So there you go. Right? Okay. All right. So another extracellular appendage that some types of bacteria can have are fimbriae. Fimbriae I like to think of as little fingers that the bacteria can project out. Fimbriae, just like your fingers help you grasp onto things, fimbriae for the bacteria help them grasp onto their environment. So you'll see the bacteria that form plaque in your mouth, on your teeth, they're going to have fimbriae. Bacteria that need to stick into your digestive tract, they're going to have fimbriae. And again, it's to aid in attachment to a surface. All right, a specialized type of fimbriae is called pili or pilus. This specialized fimbriae is going to allow specifically for DNA to be transferred from one cell to another. Ooh, that colors. Has the fimbriae, so he is the one producing the pilus to his friend. And it's along this pilus he'll send likely a little piece of DNA called a plasmid, and that plasmid will likely contain instructions for making fimbriae. And this is one of the ways bacterial cells can also transmit antibiotic drug resistance, right? You're transferring DNA from one to another. All right. So we've covered fimbriae, we've covered the sex pilus, we've covered flagella, and we've covered capsules. Now moving our way in, we get to the cell wall. The function of a bacterial cell wall is to prevent lysis when it finds itself in an unfavorable environment, whether it's the stomach or the body, or not the body, but like outside of the body. Cell wall is crucial in helping prevent lysis. In bacteria, the cell wall is composed somewhat of peptidoglycan. All right, and we'll talk more about that in a second. It's the basis, and it's the amount and arrangement of peptidoglycan that is the basis for Gram staining differences. Okay, so here is the molecular structure of peptidoglycan. It's a polymer of repeating disaccharide of NAG and NAM units. You don't need to know what NAG and NAM stand for, but just in case you're curious, NAG stands for N-acetylglucosamine, and NAM stands for N-acetylmuramic acid. So it's repeating combinations of these disaccharides that form the basis of our peptidoglycan cell wall. And this bond right here, it can be a target for antibiotic activity, right? Because if we're saying this structure, this building block of peptidoglycan is only made by bacteria, this if I can prevent this assembly, that's something that would only affect bacterial cells. Your cells should be fine because they're not making this molecule, and it turns out that's actually how penicillin works. All right, so we have our gram-positive bacterial cells, right? So you have, think about it, if you have all the bacteria, right, in existence, you have a certain type that are going to have these gram-positive cell walls, and another type that are going to be gram-negative, and we'll talk about some of the other different types, just keep in mind, right? So we're talking about a group of bacteria that have a certain cell wall structure, and these are called gram-positive bacteria. Gram-positive bacteria have a very thick layer of peptidoglycan. So what you're looking at here, the brown portion here is the layer of a huge chunk of peptidoglycan. And if we go ahead and zoom in on that, you can see the repeating NAG and NAM subunits, right? There are other components here in this huge chunk of peptidoglycan that are going to be important for us later on. Lipoteichoic acid, right? Lipid A spread throughout. Lipid A, when your body sees that, it knows immediately there's a bacterial present. Teichoic acid is another one. Lipopolysaccharide. All of the ones I've circled are triggers for your immune system. If your immune cells see these components, they can either trigger fever, trigger inflammation. It's a way to let your body know that there's a bacterial infection so it can recruit other white blood cells to come and help fight the infection. So important parts here, just as an overview, gram-positive cell walls have a very thick outer layer of peptidoglycan. On the inside, they have the plasma membrane, right? That's this yellow portion here. All right, gram-negative bacteria also have peptidoglycan in their cell walls, but it's only a small portion of their cell wall, and it's actually sandwiched in between two lipid layers, right? So you have the plasma membrane on the inside, a thin layer of peptidoglycan, and an outer lipopolysaccharide layer of lipid. Okay, so again, this is just a little compare and contrast for you guys. So this is our gram-positive cell wall, thick layer of peptidoglycan with teichoic acid present. Gram-negative cell wall, thin layer of peptidoglycan sandwiched in between two lipopolysaccharide layers. All right, so this brings us back to gram staining. So we talked a little bit last time about the actual lab technique of gram staining, the dyes you add. So now we're going to tie in the cell biology with that actual technique and result. So how to tell the difference between gram-positive and gram-negative cells, right? So just to remind you guys, the first step is the application of our crystal violet, which dyes everything purple, right? The second step is the addition of our iodine or our mordant, which enlarges the crystal violet particles and will prevent their removal from gram-positive cells. So the third step is an alcohol wash, which is going to decolorize our gram-negative cells. Our gram-positive cells will retain the purple color. And the last step is the addition of saffron, which is our counterstain. So the cells that had the dye removed in the previous step will now be able to take up the gram-negative dye. Alrighty. Okay, this is an example of what a gram-positive organism would look like after a gram stain, and a gram-negative would look like after a gram stain. Right? These guys are in chains. These guys are bacilli. Just to go over the terms we used before. So gram-positive are purple and gram-negative would be red. Okay, this just explains the mechanism. This guy is just an explanation of what is happening at the molecular level to cause the two differences we see in the gram stain. So for both gram-positive and gram-negative cells, so this is step 1 and step 2. Right, step 1 and step 2 here. Crystal violet iodine crystals form within the cell wall, right? For gram-positive cells, when I add the alcohol, so this is step 3, alcohol is going to dehydrate that really thick outer layer of peptidoglycan and trap the crystal violet iodine crystals so that they aren't able to leave the cell wall. When I go ahead and add alcohol, so again, this is step 3, in gram-negative cells, the alcohol is actually going to dissolve the outer lipid layer but leave the peptidoglycan kind of intact, and because it hasn't collapsed, it allows the crystal violet iodine crystals to be washed out, right? And so then step 4 for both of these would be the addition of the safranin or that counter stain, and because the gram-negative cells are clear, they'll be able to absorb the dye. Gram-positive cells still have that purple crystal violet iodine, so that's the color they'll stay. Some other differences between the cell walls of gram-positive and gram-negative cells. Gram-positive cells can be disrupted by lysozyme. Lysozyme is actually an enzyme that your cells produce that specifically cleaves and inhibits peptidoglycan. So it's actually a component of your sweat and tears. Lysozyme cleaves apart that outer wall of bacterial cells. Because they have such a huge amount of peptidoglycan, gram-positive cells are also going to be penicillin sensitive, so they're going to respond very quickly to the antibiotic penicillin. And again, the mechanism of action of penicillin is to disrupt those repeating subunits. So because the cell wall of gram-positive cells is so full of peptidoglycan, it turns out to be a very good inhibitor. Gram-negative cell walls, this outer lipid layer is actually a toxin. It contains toxins. So often when you talk about a bacteria or a bacterial infection causing they're causing massive fever, massive hemorrhaging. Often it's caused by an endotoxin on the outer surface of that bacterial cell wall. Gram-negative bacteria tend to respond a little bit better to tetracycline antibiotics. Tetracycline inhibits protein synthesis, so it's going to be inhibiting the bacterial ribosome, which we'll talk about in just a second. Again, because they don't have as much peptidoglycan, so it may be a better option when treating gram-negative bacterial infections. Okay. So in addition to gram-positive and gram-negative bacteria, there are other types of bacteria that we can find that cause disease. So that's what this slide is talking about. We have some bacteria that have atypical or not common cell walls, so that means that they don't fall into the gram-negative or gram-positive category. One particular category are going to be acid-fast bacteria. These guys have a similar structure to gram-positive cells, but they have an outer lipid waxy layer composed of a substance called mycolic acid that is bound to that outer peptidoglycan layer. And so because it has this outer waxy surface, it's actually not going to gram stain very efficiently. So the two species of bacteria that have this waxy layer that are considered acid-fast bacteria are Mycobacterium and Nocardia. Mycobacterium we'll talk about more than Nocardia. Mycobacterium tuberculosis is the bacteria that causes tuberculosis, and there are a couple other members of that family that can also cause respiratory disease in people that we'll talk about later in the semester. But when you see the term acid-fast, that's what's happening. The acid-fast bacteria are actually the pink groups of bacteria here. So acid-fast, pink, so this is your Mycobacterium. Two other groups of bacteria that lack your typical cell walls are mycoplasmas. Mycoplasmas are bacteria that actually lack cell walls entirely, so they just have that plasma membrane. They have additional sterols in their plasma membrane that help them to maintain integrity. Mycoplasma pneumoniae is the bacteria that causes walking pneumonia. So if you've ever had like walking pneumonia or had like a really persistent cough that just doesn't go away, possibly you have an infection of Mycoplasma pneumoniae. And then we have archaea. Archaea are actually not a type of bacteria. They are a completely separate group of ancient organisms that are found in the environment. They don't cause disease in humans because they're found in such extreme environments. Humans don't really come into contact with them, and they haven't adapted to survive within the environment of the human body. So they can either be wall-less or they can have cell walls composed of pseudomurin. It's similar to peptidoglycan, but structurally different, so it's lacking that NAM subunit. So there you go, archaea. All right, so we covered cell wall differences. Now we're on to plasma membrane. The plasma membrane is very functionally similar between prokaryotic and eukaryotic cells. It's a phospholipid bilayer with embedded proteins and channels for transport. So here's an image, and again, it looks very, very similar, if not identical, to the images you may have seen of eukaryotic cells. Okay, just like the plasma membrane of eukaryotic cells, the plasma membrane in prokaryotic cells is selectively permeable, which means it controls what materials pass back and forth. Within the plasma membrane of bacterial cells, we find the molecular machinery, the enzymes that are required for producing ATP. Right? In certain types of bacteria, we also find photosynthetic pigments on folds of the plasma membrane called chromatophores or thylakoids. It's a similar term used to describe components of, components of the chloroplast. So what's actually happening here, so you have the cell wall, the plasma membrane, and then it's like making little loops, right, around these structures, to increase the surface area, so I can put a lot of enzymes on here that will have different functions. So in this case, these enzymes found on this plasma membrane are going to be to absorb sunlight. And it's similar, so when you think about the enzymes for ATP production, if you think about how the mitochondria is arranged, and along the inner membrane they have those enzymes, a similar concept. There are special proteins that may be present in the plasma membrane of bacteria called aquaporins. Aquaporins are specifically channels in the plasma membrane that allow for the rapid transit of water across the plasma membrane. Okay. Aquaporins, along with the cell wall of the bacteria, help to control the integrity of the bacteria in different osmotic conditions. So in the human body, let's say, you can have an isotonic solution, right? So there's generally the same number of solutes in the outside as there are in the inside, and you have just kind of passive diffusion of water occurring. If the bacteria finds itself in a hypotonic solution, right, the water is going to rush in to try and balance out the solute concentrations, and without this rigid cell wall to help it maintain its integrity, the water would rush in with such intensity that it would actually cause the lysis or rupture or bursting of this bacterial cell. It is also possible for the bacteria to find itself in a hypertonic solution. In this instance, water is going to leave the bacterial cell. It's not going to rupture, but it will deform its shape. And if you did this with red blood cells, you'd see the red blood cell would be looking kind of like this. But because of the cell wall, it's able to maintain some of its integrity. Why is this important for us? Again, the presence of the bacterial cell wall helps to prevent lysis. All right, now we're moving to the real true interior of the bacterial cell. This is where we find the bacterial chromosome. Again, it's circular and it lacks histones. Histones, so if we have our DNA, coiled, in eukaryotic cells, so in our cells, there are special packaging proteins that help coil the DNA and help it wrap and maintain its shape. Those special packaging proteins, again, because we have so much DNA, we have several chromosomes, several pairs of chromosomes. Each of those are composed of massive amounts of genetic material. Histones are really essential for helping eukaryotic cells package and organize their DNA. So in prokaryotic cells, they do not have these packaging histones. They have less genetic material to deal with, so they just kind of plop it all together. Where they're plopping it in the cell, it's not a nucleus, it's a nucleus type area, so we call that a nucleoid. But again, there's no nuclear envelope that separates that genetic material from the rest of the cytoplasm. All right, so ribosomes. Just like in your cells, the genetic material that we have needs to be transcribed and then translated into proteins. The ribosomes are the functional units that are going to be doing that translation, but there are some differences between eukaryotic and prokaryotic ribosomes. But again, the overall function is the same, that of protein synthesis. The ribosomes in both cases are composed of ribosomal RNA, so that's what this little R stands for. It's ribosomal RNA. As opposed to the other types of RNA, which are we have the tRNA, and then we have the mRNA, which is the messenger RNA. Oh my goodness, my writing. So we have T for transfer, and they are bringing the amino acids to the ribosome, and the messenger RNA is what is transcribed from the DNA. In prokaryotic cells, the ribosomes are called 70S ribosomes. You can think about this S as standing for size, so it's like size 70. That's actually not what the S stands for. It's a sedimentation factor, which is why you can have something that's 50S and something that's 30S add up to 70. So it's not a typo. It's just because the S doesn't actually stand for size. So sedimentation factor is talking about, if I spin, a sample of ribosomes super, super fast, like in a centrifuge, at what point do those centrifuge, do the ribosomes begin to pull out of solution or sediment? It's talking about that. So we have a large subunit, which is a 50S, and a small subunit, which is a 30S. And if they are in a complex together, we have this 70S ribosome. And this size of ribosome is unique to prokaryotic cells. All right, so it is possible for some bacteria to also have inclusions or things that are found in the cytoplasm that aren't ribosomes or genetic material. Names for these inclusions, we can call them granules, or we can just call them inclusions. The function is for energy storage. So sometimes we can also have iron particles stored here. So in this case, these are magnetosomes. We can have ones that store different gases that the bacteria might be producing as a byproduct. The magnetosomes may contain iron oxide, which help destroy hydrogen peroxide. And some bacteria can actually orient themselves to magnetic north, which is kind of cool. But so there you go. So sometimes you can have other random stuff appear. But again, these are not separated from the rest of the cell with plasma membrane, so they're not organelles, right? So, because bacteria do not have organelles. Okay. All right. Some bacteria also have the ability to form endospores. Endospores are kind of like bacterial seeds, right? So if you think about plants and how plants produce seeds, they're basically ready to go. They're able to germinate once they find themselves in the right environment. Bacteria produce endospores, and they are functionally very similar. So their endospores are considered resting cells. So again, think about a seed. So it's not actively doing any metabolism, so it doesn't need any nutrients or water or anything like that. But once it finds itself in the right condition, like a human body, it is able to germinate and then would be able to cause disease. In this endospore state, they're resistant to desiccation, heat, and different chemicals. Two genuses of bacteria that regularly form endospores. So here we go. So for bacillus, think about anthrax, right? The reason why you're able to mail anthrax to someone and it stay infectious is because it's in the endospore state, right? Usually bacteria, in order to transmit it from person to person, you need pretty close contact. But bacillus can form endospores, which is why you can send it in the mail. Clostridium we'll talk about. There's a bunch of different types of Clostridium. It can cause botulism, gangrene, tetanus, Clostridium tetani. Okay, and so again, you're stepping on a rusty nail. You can get this bacteria because it lives in the soil. All right, so sporulation is the process of the bacteria forming the endospore, and germination is the process of that endospore returning to an active state. Right. This shows you a little bit more detail about the process of endospore formation. So again, you have a bacterial cell, realizes times are getting tough, maybe it finds itself in the soil or what have you. So instead of actively dividing and forming two new bacterial cells, what's going to happen here is it's going to start to encase one of its duplicated bacterial chromosomes in additional layers of plasma membrane and cell wall, so that when the parent cell eventually dies and disintegrates, you have this really encased and protected genetic material. And again, when it receives the right signals from the environment, it is able to germinate. Okay, so now let's talk about eukaryotic cells. And if you need to pause the video and take a little break, now's a good break time. But don't forget to come back. All right, so most of this is going to be review from general biology, where you talked pretty extensively about plant and animal cells. So I'm going to go through this pretty quickly just to refresh your memory. All right, again, working from the outside in. So eukaryotic cells can have flagella, or they can have cilia. Cilia are little tiny extensions. Kind of think about rowboats, like all the little oars kind of going together, or those crazy comb jellyfish that go like like spoodle all over the place. Cilia. They can also have, so in their flagella, we find microtubules. Microtubules are composed of tubulin. In a flagella, we have 9 pairs, surrounded by 2 central microtubules. But I'm going to go ahead and say you don't need that information. All right, so surrounded by a plasma membrane, right? So it's not the filament with the hook and the basal body, right? That was from bacteria. So this guy is just extension of the plasma membrane. Cell walls can be present in eukaryotic cells, especially when we talk about plants and fungi. Specifically when we talk about microbiology, we're referring to algae of the plant group. And the cell wall of algae is composed of cellulose, which is a type of sugar that plants make, and they use it in their cell walls. And fungi also have cell walls, but again, like fungus are not plants, so their cell wall is composed of a different material. It's chitin. So if you see chitin, in the question, you're talking about a fungus. If you see cellulose, you're talking about a plant. If you see peptidoglycan, you're talking about bacteria, right? Some animal cells can also secrete that glycocalyx, and in bacteria, the glycocalyx can be formed into a capsule that protects them from your immune system. And some animal cells that are unicellular just can form a glycocalyx to help them adhere to different surfaces. All right, the plasma membrane is also selectively permeable, and because there's no cell wall, we can do specialized transport like endocytosis and exocytosis. There are 2 types of endocytosis we will see. Phagocytosis is going to be important when we talk about the immune system, and this is pseudopods or little false feet extending up. Right here, extending up to come and surround and engulf the foreign particle, and then when it's completely engulfed, they take it inside the cell, and it forms a phagosome. You can also get small pinocytosis, where the membranes fold inwards, but that is going to be less relevant for us. And again, phagocytosis is important. We'll talk about that when we get to the immune system. Okay. Well, see, moving quick. All right, so ribosomes, function is the same, protein synthesis. In eukaryotic cells, we have ADS, So again, it's a larger, more complex ribosome. Usually they are found attached to the rough endoplasmic reticulum, or RER. They can also be found freely floating around the cytoplasm. All right, so they do the bulk of the work for eukaryotic cells, the ADS. However, 70S ribosomes, which are bacterial in origin, can also be found in eukaryotic cells, but specifically in the chloroplasts and in the mitochondria, which are organelles, and we'll talk about it just coming up in a second. There's a current theory, the theory of endosymbiosis in biology, that states that chloroplasts and mitochondria, these two organelles found in eukaryotic cells, are actually, they evolved from engulfed bacteria. Right? So it makes sense now, right? And one of the pieces of evidence that supports this theory is that we can find bacterial ribosomes, these 70S ribosomes, in these organelles, right? So that's suggesting that at one point, these guys were free-living bacteria that had their own protein machinery, 70S. So there you go. So technically, right, both ADS and 70S can be found in an entire eukaryotic cell, but 70S are specifically found in those organelles. Okay. Organelles that I'm not going to go through in detail, but in a eukaryotic cell we have the nucleus. That's what gives it its name, eukaryotic, or true nucleus, and it's where it has a nuclear envelope surrounded by nuclear pores. It has an endoplasmic reticulum network. We have smooth and rough varieties. Rough usually is encased in or surrounded by ribosomes. The smooth ER is going to have different things like cholesterol synthesis and stores various materials for the cell. Golgi complex is going to be a site of protein modification and maturation for the cell. So if you can imagine proteins being synthesized by ribosomes, and they get shipped to the Golgi body where they get further modified. And then we have the lysosome, which contains digestive enzymes and storage vacuoles for support. Other organelles. The mitochondria, which is involved, it's the powerhouse for the cell. It's producing ATP. We'll talk about that in more detail. Plant cells have chloroplasts. Animal cells do not. Both animal and plant cells have mitochondria. Some cells also contain peroxisomes, which is very similar to a lysosome, only instead of digestive enzymes, it has the ability to degrade hydrogen peroxide, which can be toxic to cells, and then centrosomes, which are going to assist in cell division. So this is just a little bit more about the mitochondria, which we'll talk about in a second. So we have a double membrane, right? And the inner membrane is highly folded, and this is similar to some bacteria, right? So bacteria would have a cell wall here, and their plasma membrane would be highly enfolded with different types of molecular machinery on there. Chloroplast, same thing, double membrane. So we have this outer function similarly to a bacterial cell wall, and then the inner plasma membrane is highly folded. A stack of folds is called a granum, and the individual membranes are thylakoids. Similar terminology is used between chloroplasts and some bacterial cells. All right, so the piece de resistance, right? The theory of endosymbiosis, or the endosymbiotic theory. So this theory states that life first evolved, right? The first living organisms were probably bacterial in origin, or looked like archaea, around 4 billion years ago. The planet itself was only formed about 4.5 billion years ago, so, just to give you a frame of reference. Eukaryotic cells first came on the scene, or we have evidence for them first being on the scene, 2.5 billion years ago, right? So a lot of time has passed, obviously, since then. But so this, the theory states that eukaryotic life evolved from a larger prokaryotic cell that engulfed smaller, specialized prokaryotic cells. And these smaller, specialized prokaryotic cells eventually came to reside within the larger eukaryotic cell, right? So looking at this image, we have an early ancestral cell. We have, from this early first life, we have bacteria, we have archaea, and then we have these larger cells that don't have cell walls. They have plasma membranes, and they probably survive by eating other smaller bacterial cells. Because of this weird, funky plasma membrane, it likely at some point would encase its genetic material, right? It turns out it's like a little bit more stable that way. You don't get as many mutations. And at some point, this cell is pretty large and, again, survives by engulfing other cells, but he can only be so large, right? At some point, he engulfs a small bacteria that's really, really good at producing lots of energy if you give it some sugar. So what happens is they form a mutually beneficial symbiotic relationship, and over time, they evolve together. Right, and if you look at the way mitochondria divide, they're still dividing, even though they're inside this larger cell with binary fission. They still have bacterial ribosomes, and they also have some bacterial DNA. Right, so there's a good evidence to support this, right? And so we have these guys going off to form animal cells, and then we have another branch at some point that does this little trick again and engulfs a chloroplast, and these guys will become your plants, right? So evidence for this theory, again, it's the presence of mitochondria and chloroplasts within eukaryotic cells, and the pieces of evidence that support this theory, right? We have first piece of evidence, both mitochondria and chloroplasts have their own DNA, and not only do they have unique DNA within themselves, it's circular DNA, which is the same shape, and it lacks histones, the same shape and structure as bacterial DNA. The second piece of evidence is that they have their own ribosomes, right, to take that DNA and translate it into proteins. And not only do they have their own ribosomes, but those ribosomes are bacterial in nature. They are 70S ribosomes. Both mitochondria and chloroplasts don't divide when the cell undergoes mitosis. They kind of divide on their own schedule, and they divide via binary fission. And they both have double membranes, and they have unique enzymes. The double membranes, right? So again, if this guy had a cell wall and then the host cell degraded it, he's still going to have an additional outer membrane from the host cell, right? And the same thing here. So double membranes is kind of a little throwback to like, oh, it probably was engulfed at some point. Cool. All right, so that's it. I know it was a big lecture, pretty dense. Hopefully, a lot of it was review from previous courses you've taken. And if you guys have any questions about any of this material, please feel free to shoot me an email, or we'll probably be discussing some finer points of this in lecture as well. So I hope you enjoyed it. Hope it wasn't too horrible, and I will see you guys in class.